So today I'm going to be telling you how DNA replication works. Hope you enjoy. So step one is the DNA helicase opens up the strands. So we're untwisting the pipe cleaners. And the DNA helicase opens up the strands to create a replication fork, which is a Y-shaped structure which forms when the DNA is split into two strands by DNA helicase. DNA helicase is an enzyme which is responsible for unwinding and unzipping the DNA strands during replication. Alright, so this is the lagging strand in this experiment, and this is the leading strand. And with the leading strand, this is the 5' end, and this is the 3' end. And with the lagging strand, this is the 3' end, and this is the 5' end. So we start with the leading strand, and for the leading strand, we're going to add an RNA primer at the top where the 5' end is. And the RNA primer allows the DNA polymerase to start building from the 5' end to the 3' end. And the RNA primer is at the top because that's where the 5' end is, and it needs to build down towards the 3' end. All right, so this is the DNA polymerase in our experiment, and DNA polymerase is the enzyme responsible for adding complementary nucleotides to the new DNA strand during replication. This is the leading strand, and the purple pipe cleaner will be creating the complementary strand from the 5' end to the 3' end. Alright, so now the DNA polymerase has been added and fully wrapped around the original strand, but it has to stop at the replication fork because it has nowhere further to continue adding polymerase. Now that DNA polymerase has been fully added to the leading strand, we can do the lagging strand. So for the lagging strand, it has to be done a little differently because the 5' end and the 3' end are swapped. So instead of putting the RNA primer at the top, like on the leading strand, we're going to put it at the bottom, at the 5' end there. So the RNA primer is added at the bottom, and now DNA polymerase now has to be added in the opposite direction, but still in the 5' to 3' end. So, and this is called an Okazaki fragment. Okazaki fragments are short sections of DNA which are formed on the lagging strand during replication. DNA polymerase can only add nucleotides in one direction, so these fragments are created and later joined together to form a complete strand. So the yellow pipe cleaner represents the DNA polymerase, which is being added to the original lagging strand. So now that both strands have fully added their DNA polymerase and RNA primer and are finished, now we need to open up the replication fork even more and to get closer to finishing the complementary DNA strands. So while all of this is going on, there are two problems which are being prevented. One is over twisting of the strand. The topoisomerase fixes. And topoisomerase is an enzyme which helps prevent DNA from becoming too tightly coiled or twisted during replication by making temporary cuts, then rejoining the strands. And the second problem is the strands coming back together, which single-strand binding proteins fixes. Single-strand binding proteins are proteins that prevent the separated DNA strands from reattach after being unwound by helicase. Okay, so now we are unwinding the DNA some more so that we can add more DNA polymerase and get closer to finishing the strands. So the DNA is still going to be added on the leading strand continuously by just wrapping and wrapping around. So the purple cleaner here doesn't have much to do except just keep on wrapping until it reaches the end of the replication fork. However, since the 5' end and the 3' end are swapped on the lagging strand has to put another RNA primer at the bottom of the fork and then work its way up to where the previous primer had been, which creates another Okazaki fragment. Okay, so now we're adding RNA primer on the 5' end of the lagging strand, and then we're going to add DNA polymerase, which we're doing now. And now we're going to add DNA polymerase, which is going to work its way from the 5' end, which has the RNA primer on it, to closer to the 3' end, which has another RNA primer on it. So now DNA polymerase is wrapping around the original strand on the lagging strand. Okay, so now that we understand the concept, we're going to keep on wrapping and adding polymerase and RNA primers until we reach the end of the strand, and then it should be finished, but then there's more steps after that. Okay, so now we have separate strands, but they are not finished yet. So they each contain one original parent strand, which is why they're complementary. And a complementary strand means that they are semi-conservative. And semi-conservative is a method of DNA replication where each new DNA molecule consists of one original strand and one newly synthesized strand. But now that we have two separate strands, there's a problem. Problem is that there is still RNA primer on the strands, which we need to remove. On the leading strand, it's much easier, and exonuclease removes this RNA. And exonuclease is an enzyme which removes RNA primers from the DNA strands. On the lagging strands, it's a lot more complicated because there's a lot more RNA primers that exonuclease has to remove. After exonuclease removes the RNA primers, DNA polymerase fills in these spaces which the RNA primer possessed beforehand, and it fills them in with new DNA. Then after this new DNA has replaced the RNA, it's kind of like not really connected to the other parts of the leading and lagging strand. So DNA ligase comes in and puts these fragments together and connects them all. After the DNA ligase goes through and connects all the strands, the two new semi-conservative strands are finished. This is the lagging strand. And the lagging strand is the strand which is synthesized discontinuously in short Okazaki fragments during replication. And this is the leading strand, which is the strand that is continuously synthesized in the 5' to 3' direction during replication, and the one that moves in the same direction as the replication fork.